But I'm going to just spend the next few minutes talking to you about a recent paper that has just come out, and thanks very much, Marta, for giving a good lead in to some of this work. It's been a really busy year for three papers to actually come out on this topic. But I actually think that's great, because it's almost as if there's kind of independent replication and independent kind of support for the method coming out at the same time. So I actually think that's really good. Our eDNA expert that we worked with on this project most currently is Dr. Michaela Hellstrom. So some of you may have worked with her before. She's based in Sweden. She has a company called Mix Research. Okay, so the last thing I'll just say is that most of us who work at WWF know, and at least in the Arctic program, we don't much have the opportunity to actually be leading or doing a lot of research in many of our jobs. I think maybe there's some of us that are lucky enough to do that. But most of the time, you know, we're a science-based organization. So most of the time, we're looking for collaborators such as the lecturers and researchers that we've heard on the call today to be actually working with us, or we're supporting their research to help fill knowledge gaps. So that we have the best science to inform our positions on what we think needs to be done for conservation. This project was a little bit of an exception. We actually were able to kind of co-lead this project over the last decade. So we're really glad that we've now got this method published in the scientific literature. Okay, so we've heard a lot today already that it's really important to be understanding species responses to climate change and other stresses, enabled to monitor them in the most basic way, to understand how many animals we have, how those trends are changing over time, and also to answer a lot of other questions about population demographics, relatedness, relationships, evolutionary capacity, which genetics can help us with. And unbelievably, I think for a lot of people who believe that polar bears are really well studied, which in some respects they are, for 10 of the 19 polar bear subpopulations across the Arctic, we actually don't have basic information on the number of bears. So we have a global estimate of between 22 and 31,000 polar bears, but for 10 of those 19 subpopulations, we don't know how many bears we have. So we're missing some basic information to help inform management and conservation efforts. It's important to mention here, and as it's been highlighted, the method that has been developed here, it's a non-invasive method. So there's no need to even see the bear in order to collect the snow steps. And this fits well as an additional tool in the toolbox, because there are some concerns from some Arctic Indigenous communities about the effects of research techniques that are used to gain tissue samples. And the methods that are used for polar bears currently include capture and sedation, but we also know that at that time, while a tissue sample is taken, there's a whole lot of other information that's collected on the bears, and often satellite trackers are fitted to bears, so we get a lot of other information. But the other main method for collecting tissue samples is biopsy darting, often from helicopters. So there are some concerns about the effects of different sedatives and also disturbance from helicopters on polar bears, and the possibility for habituation. So there has been more recent work done on hair and feces, which can also be quite hard to find for polar bears. So really it's helpful for us to have more tools in the conservation toolbox to help us collect samples of polar bears. We've also heard from, for example, Russian colleagues, when they've been out doing aerial surveys to try and count polar bears, that in some parts of Russia, they've seen so many footprints and hardly any bears. And so, you know, if those footprints were able to be collected, that might help with some insights to be able to actually create some sort of baseline database for genetic mark recapture. Okay, as Marta said, this particular project, well, as Marta said, it's for a long time actually, researchers have been able to access mitochondrial DNA from snow steps, but the real challenge that has remained until recently is getting access to that nuclear DNA, which is not really unexpected, because mammalian cells just have far more mitochondrial DNA in them than they have nuclear DNA. But mitochondrial DNA doesn't give us individual identities. So it's really the nuclear DNA that we're looking for if we want to get to that individual identity. So for WWF, we've come to where we are now from about 10 years of discontinuous collaboration. So we actually started work with a researcher at 𝐨𝐫𝐤𝐬𝐡𝐨𝐩 in the early 2010 decade, and there they were able to actually isolate mitochondrial DNA from snow steps of polar bears. Then we just had a bit of a lapse where people left and came and left and changed over and that kind of thing. And so our most recent collaboration has been with Dr. Michaela Hellstrom for about the last 5 years. And so, yeah, we've been finally able to isolate nuclear DNA from snow steps of polar bears and a couple of other species, which I'll get to. So this collaboration with Michaela involved trials in captivity with polar bears, lynx, and snow leopards. And that was also collecting at the same time other materials as positive controls, such as saliva, hairs, and nasal mucus. And then there was also some field collection and analysis of lynx and polar bear snow steps. And just to mention that the lynx portion of this work, I'll do my best to explain because I'm sure that's of interest to this audience, but that work was actually Michaela's work that was done previous to the collaboration with us, but we included it in the paper. And the protocol that's in that paper, which we used for polar bears as well, was really kind of founded on lynx and built on polar bears after that. And so that collection and analysis was done, and then we're up to the stage now where we've just had those published. Okay, so how is it done? So we, for us, I'll talk mostly about the polar bear component, but a little bit about the lynx as well, because that's in the publication. So the first thing to do is find footprints. In Alaska, we did this with the expert assistance of Alaska Native hunters, who were fantastic at everything, understanding the weather, understanding the environment, understanding where the polar bears might be, how safe we were around them, and that kind of thing. So that was invaluable assistance that we had there. For lynx, I understand that Michaela worked with trackers, hunters, and local administrative board staff around central and southern Sweden to collect those footprints. And they found that for lynx, it's not uncommon for multiple animals to actually walk in each other's footsteps. And they obviously wanted to avoid that. And the trackers were actually able to find that point where the tracks deviated and then start collecting the snow from then. And we actually find the same thing with polar bears, not that they necessarily walk in each other's footsteps, but cubs will stay with mothers for about the first 2 years of life. And polar bear mothers can have two cubs at a time. So that's the kind of the main thing that you want to do, is make sure when you're sampling a footprint that you're sampling one footprint and hopefully not multiple, the footprints of multiple animals, because it then, you know, virtually becomes incredibly difficult to separate out individual genotypes. So as Marta also shown in pictures, we collected a thin layer of snow from the prints. And so that was kind of all of the print that we could see, including the main foot pad and the toes. Using really simple tools. You can see there I'm just using a serving spoon and a paper bag, a plastic bag. But having said that, the tools need to kind of be as sterile as they can. We work to collect as many prints as possible because there's such small amounts of DNA. The more you can collect, the better. And it actually didn't seem to take us very long to do that, which was good. And that collection, which I think Tomasz also mentioned, collection can be done by non-experts, and I'll get into that in a second. That we then melted and filtered the snow, capturing the cells containing the DNA in the filters. So basically that takes you from, if you look at the size of the bag there, it takes you from trying to freeze and transport a large bag of snow, or even 2 large bags of snow for, you know, one sample effectively, to very small filters. So that was much easier for transport. And then the DNA was extracted and analysed using individual level genetic markers. And here we used microsatellites just to see if we had accessed the nuclear DNA. And obviously, as mentioned already, that requires molecular and eDNA expertise and facilities to do that. I should mention though, for steps 2 and 3, we actually did one field collection trip with polar bears earlier in 2023 off a Coast Guard vessel off Svalbard, and we actually were able to set up a temporary lab on the vessel where we were able to just sterilize the equipment and turn it around between sampling, and then also melt and filter the snow there. So there are steps that can be done with quite rudimentary setups. Okay, so in terms of success, which has been talked about a little bit, so we successfully accessed the nuclear DNA from the snow steppes in polar bears and lynx from captivity in the wild, and also from the one snow leopard that was sampled in captivity. So Marta, I should mention, and Marta, you can talk about this a little bit more if you want to when I'm done, but Marta, your paper went further than ours did. So we were not looking specifically to get to individual level identification. To do that, we would have required screening all individuals, probably in at least triplicate. We would have required a much larger panel of microsatellites than we used, but we were basically just seeing if we could access nuclear DNA, and we were using a subset of microsatellites to do that. So we sampled 24 wild polar bear tracks, and of those, most of them we got nuclear DNA out of. So for most of them, we actually got at least one microsatellite genotype. For just over half of them, we had 5 loci that we screened polar bear samples at, and we got 13 successfully genotyped at 5 loci. And of those, 11 were unique genotypes with 2 identical genotypes. So probably the 2 identical genotypes was one bear, because the 2 samples were taken one day after the other. So probably of the 13 samples, we got genotypes from 12 bears. And then Michaela's earlier work included sampling of 44 wild lynx tracks, and we had a lower success rate with lynx than we did with polar bear. And we also had different success rates when Michaela had untrained personnel collecting versus trained personnel. And by that, trained personnel were individuals who were provided with written protocols, but then they completed a 1-hour theoretical training. They observed sample collection by experts first, and then they collected samples under the supervision of the trainers. So when we say trained personnel, that's what we mean. And that maybe gives you a bit of an idea of what it might take to get volunteers to be collecting samples. And so untrained personnel and volunteers had access to the written protocols, but they didn't receive hands-on training prior to collecting the samples. Okay, and then just finally, I've talked a lot about polar bears because I guess that's mostly what I work on, and that's what we'll be kind of following through. And that was sort of our initial WWF's initial involvement and start up with this work. But it was really good that we were able to publish on all 3 species, because I think that there are applications for other species that live in snowy environments here. And as Marta's group's shown, and there's others as well, I think there's really good potential to be looking at this technique further. We did find that collection could be done by trackers, trappers, and other non-genetics experts with some basic training. That's really relevant and useful for us in the Arctic, where there's a lot of Indigenous communities living in coastal areas and who have polar bears coming through all year long. So it really adds potential for greater spatial and temporal sampling of populations, which is important. The other thing to note is that the snow can be stored frozen. So if you've got people collecting, but they can't filter the samples, for example, or you don't feel confident asking them to do that, the snow can be frozen and then shipped even frozen to a lab. It just means that it's a lot more bulk and a lot more freezer space needed to do that. As Marta mentioned, for us, we're not talking about this as being an alternative to other tools that are out there to be sampling animals for population genetic studies, more just that it's an additional tool and a way of collecting additional samples towards those genetic studies. And so my advice would be to collaborate with those who are already doing those studies, because they will probably already have the markers and the primers and the genetic lab set up to actually do that analysis and take in those other samples. The only warning, of course, is that because the samples contain such trace amounts of DNA, it's generally not a good idea to be extracting DNA from snow steppes in the same lab that you're extracting DNA from tissue samples or something like that, unless you've got really good protocols. So generally you would presumably be collaborating with someone who's working with your species who has access to an eDNA facility or a lab or something like that. A few considerations that we found so far through most of Michaela's work. So I think she found that you can still extract DNA from snow steps that are 2 to 3 weeks old. We were quite unsure about how UV would affect the DNA, because UV is really, really bad for DNA, but basically kills it. So, but we didn't find that was too much of an issue, but probably some more work is needed there. We found that it works in some snow types better than others. So kind of depends on the weather, depends on the conditions. We even noted that polar bears seem to put less of their foot pads down when it's really, really cold on the snow. So it's not, I think it's not always going to work is the point, and it depends on a lot of factors. Generally, we found the more steps collected, the more DNA you get. Maybe animals with small feet or foot pads might need a greater number of steps collected. And you need to really be able to single out the steps. So if you've got animals, if you've got a lot of steps that you can't single out the tracks of a single individual, it might be sort of quite difficult to actually be able to make this useful for individual level analysis. The success rate isn't as high as collecting from tissue and skin samples, and the cost of genotyping can be higher if you need special kits. And because it's eDNA, so samples really need to be screened multiple times to confirm genotypes, because as Thomas was saying, there's high likelihood, it's pretty much a guarantee that you'll get some genotyping error because you've got degraded DNA. But I think this is something to be balanced with the relative availability of footsteps and potentially the low cost of collection relative to, for example, helicopter time for biopsy dating, which can be extremely expensive. All to say, we haven't done a specific cost comparison. And again, like the others have said, how much this costs really depends on the question being asked, the genetic markers you need to use. And, if you need 10 markers, it's half the cost of, say, needing 20 markers. So it just really depends. But I think it's definitely, it's really promising. And as I said, I'm really glad that a lot of this work has been published in this last year. So for us, the next step is really to make sure that this doesn't just sit on a shelf, but that we socialize this with the research community, and see if we have people who are doing population genetic studies on, for example, polar bears, where we can actually have snow steps be an additional sample type that can be collected and analysed. Okay, that's all from me. Thank you very much.